To sequence larger molecules, individual chromosomes are purified and broken into or larger random fragments which are cloned into vectors designed for large molecules. A 100 MB, B 100 kilobyte, C 5000 kilobyte, D 600 kilobyte. The correct answer is 100 kilobyte. Which of the following is untrue about DNA sequencing methods? A. Purified fragments of DNA cut from plasmid phage clones or amplified by polymerase chain reaction PCR. B. Clones of DNA fragments are denatured to single strands and one of the strands is hybridized to an oligonucleotide primer. C. Taq polymerase is quite heat sensitive. D. New strands of DNA are synthesized from the end of the primer. The correct answer is Taq polymerase is quite heat sensitive. Many levels of clone redundancy may be required to build a consensus map because individual clones can have A. Rearrangements, B. Deletions, C. Two separate fragments, D. Vectors. The correct answer is vectors. The combined mixture of all labeled DNA fragments is electrophoresed to the fragments by and the ladder of fragments is scanned for the presence of each of the four labels. A. Separate size, B. Separate pH, C. Assimilate pH, D. Assimilate size. The correct answer is separate size. An alternative method is to sequence all the subclones, produce a computer database of the sequences and then have the computer assemble the sequences from the overlaps that are found. A. True, B. False, C. D. The correct answer is true. Once the correct map has been obtained, unique overlapping clones are chosen for sequencing. A. True, B. False, C. D. The correct answer is true. The sequence can also be verified by making an oligonucleotide primer complementary to the distal part of the readable sequence and using it to obtain the sequence of the complementary strand on the original DNA template. A. True, B. False, C. D. The correct answer is true. Using ddATP, the resulting synthesis creates a set of nested DNA fragments, each one ending at one of the as in the sequence through the substitution of a fluorescent-labeled ddATP. A. True, B. False, C. D. The correct answer is true. When the process is fully automated, a number of priming sites may be used to obtain sequencing results that give optimal separation of bands in each region of the sequence. A. True, B. False, C. D. The correct answer is true. Which of the following is untrue about shotgun sequencing? A. When DNA fragments derived from different chromosomal regions have repeats of the same sequence, they will appear to overlap. B. When DNA fragments derived from different chromosomal regions have repeats of the same sequence, they will appear to scrutinize. C. In a new whole shotgun approach, cellular genomics is sequencing both ends of DNA fragments of short 2 kilobyte, medium 10 kilobyte, and long back or 100 kilobyte lengths. D. A large number of reads are then assembled by computer. The correct answer is when DNA fragments derived from different chromosomal regions have repeats of the same sequence, they will appear to scrutinize.